Welcome learners. Welcome to Biomindscape. Today's session focus on the core recognition machinery of immune system, that is the immune cell receptor. These receptors are very reason immune cell. It is the most important because this cell can detect, react and defend the body from any type of reaction for innate recognition and to adapt the specificity and immune receptor form the foundation of immune responses. So let's break down system by system about it. So what is a immune cell receptor? A immune cell receptor are a protein on the surface or inside a immune cell that allow them to recognize a foreign substance or receive activation of some signals and trigger appropriate immune response against this foreign substance. So there are two major types. The fixed and inherited one is a germline, that is innate immunity, and adaptive immunity is unique to each other cell clone, that is somatically recombined. So germline encoded and somatically recombined are the two major types of this. So germline encode fixed and inherited and somatic recombined is unique to each cell clone. So six major type of immune cell receptors are pattern recognition receptor, antigen-specific receptor, that are also of two types, BCR and TCR, and natural killer cell receptor and FC receptor, chemokines and complementary. These are the six major types of immune cell receptor. So immune cell receptor, two main, innate and adaptive. These are important: germline encoding and somatic recombined, and six major types of immune cell receptor. The first with the pattern recognition receptor. These are used by the innate immune cell like the macrophages, neutrophils and dendritic cells to detect the pathogen-associated molecular pattern and danger signal, that is the DAM. So they also have some other types. The types are present on the surface or the cytoplasm, and there are various ligands, important name of the ligands, which bind to the specific type of a receptor. So the toll-like receptor, it is a type of antigen-specific receptor, toll-like receptor. It is located on the surface or endosome, and the ligand which bind to the toll-like receptor are LPS, dsRNA, then CpG and CpG DNA. So these are the ligand which bind to the specific toll-like receptor. Then node-like receptor, these are the type of receptor which are present on the cytoplasm, and the ligand which bind to the node-like receptor are bacterial peptidoglycan. Then there are ring eye-like receptor which are located on the cytoplasm, and the ligand which bind to them are viral RNA. Then C-type lectin receptor, these are located on the surface, and the ligand which bind to them are fungal carbohydrate. So signaling path. So note down the table, make a flow chart of the innate immune cell receptor. So first we will write the pattern recognition receptor, that is the PRR, that recognize a pathogen associated molecular pattern, that is PAMP, and damage associated molecular pattern, that is DAMP. Then there are the various types: toll-like receptor, node-like receptor, ring eye-like receptor, C-type lectin receptor, and scavenger receptor. We have noted down the toll-like receptor, what it is used for to detect the bacterial LPR, viral RNA, etc. So it detects the gram-negative bacteria. Then node-like receptor, node 1 and node 2, they detect the bacterial peptidoglycan. Then ring-like receptor, RLR, they detect the viral RNA in the cytoplasm. Example, they detect the dsRNA from the virus. Then C-type lectin receptor, they detect the fungal carbohydrate. Example, lectin 1 binds to the beta glucan. Then scavenger receptor, they bind the motif low density lipoprotein apoptotic cells. They have an important role in this ligand binding and all these functions. So activate in the NF-kB and MPKs, IRFs to produce the cytokine and interferon. in various signaling pathway it is important. Then TLR4 bind to LPS, which further trigger the NF-kB and then produces the interleukin 1 and TNF alpha. This is one of the examples. Antigen-specific receptor like BCR and TCR. Now B cells and T cells, these are used for a specific adaptive immunity. So B cell receptor are the BCR and the T cell receptor are TCR. So here the B cell receptor have a membrane bounded IgM or IgD. They are recognized native antigen which are associated with the Ig alpha and Ig beta for the signal transduction. The signal activation in which it helps are NF-kB, MPKs and PLC, gamma and SYK. And T cell receptor recognize the peptide plus MHC complexes and has alpha and beta chain which are associated with the CD. The T cell receptor are associated with the CD3 complex with ITAMs and signals Y ZAP70, LAT and NF80. So in this CD4 and CD8 are important. CD4 bind to the MHC2 complex helper T cells and CD8 binds to the MHC1 cytotoxin T cells. Then there are natural killer cell receptor. These cells don't use in the T cell and B cell receptor. Instead they rely on the balance. So NK cells, natural killer cell receptor. So natural killer cell, they do not use the TCR or BCR. Instead they rely on the inhibitory receptor and activating receptor. Now let's know about the inhibitory receptor. So it is inhibitory. It recognizes the MHC1 molecule. It contains ITIM motifs. An example are CD94, then KIRS and NKG2A. So activating receptor are recognized. It also have natural killer cell have inhibitory as well as activating receptor as well. So the activating receptor recognize the stress-induced ligand of IgG Fc-containing ITAMs. Example NKG2D. This detect the stress ligand, and CD16 detect the IgG and mediate the ADC. So Fc receptors. There are various types of FCR receptors, that is the Fc portion of the antibodies. At that position these are present, there are binding. So Fc receptor, they are found on many immune cell like macrophages, neutrophil, NK cell. So there are Fc gamma R which bind to the IgG. Then Fc epsilon R which bind to the IgE, which is important in allergy and mast cell and basophils, and Fc alpha R which bind to the IgA. Now in that also there are various type, like they bind to the various cells and antibody. We know that Fc portion, it binds. Where does it binds? To the F site of an antibody. So here the Ig in antibody IgG, the Fc gamma R1 and R3, they have some important role in macrophages cells. In like Fc receptor bind to the IgG, express on macrophages, and it function in the phagocytosis. Then Fc gamma R3, that is CD16, it bind to the IgG, expressed on the NK cells and function ADCC. Then Fc epsilon R1, here it binds to the IgE, mast cells, expressed on the mast cell and basophil, and it functions for allergy and degranulations. This bind to the constant Fc portion of an antibody to mediate the effective function. So in Fc alpha R1, like CD89, it binds to the IgA, expressed on the neutrophils, and the function is mucosal defense. In that it is important. So we know that the IgA, it is present here in the neutrophils. which receptor of Fc, that is ER1, FCER1. So I have made a table. In that you can see how these receptors are present and the binding and their expressions on that particular cells and the functions which are given here. In chemokine receptor, these are GPCRs, that is the G protein coupled receptor that direct the immune cell trafficking, where the ligands and its role are given. So the receptor and the ligands are present and their roles in the various T cells, B cells, bone marrow, inflammation and HIV entry. So the receptor which are present in this particular chemokine receptor, as we all know that they are the GPCR that direct the immune cell trafficking. In that the CCR7, here the receptor and the ligand is CCL1 19 and 21. Its role is in T cell and in lymph nodes. So chemokine, it helps in the migration of immune cell. There is CCR7, CXCR5 and CCR5. These are some of the name of the receptors, and the ligand which they bind are like CCR will bind to the CCL19 and 21, and CXCR5 will bind to the CXCL13, CXCR4 will bind to the SDF1, and CCR5 will bind to the CCL3, 4 and 5. So what is the role of this receptor and ligand? Its role is in various cells like T cells, B cell, bone marrow and inflammation. And HIV use CCR and CXCR4 as co-receptor. Then there are complement receptor. They recognize the complement opsonized pathogen. So there are various receptor and ligands and their function, like in the phagocytes, in B cell activation, in immune complex clarity. In that the CR1, CR3 and CR2 are the receptor, and the ligand which bind to them are C3b, C4b to the CR1, and that is CD35. Whereas the CD11b and CD18, the ligand is I3b, that is in the function, it functions in the phagocytes, and CR2 like CD21, the ligand is C3d and the function is beta activation, B cell activation. So these are the various name of the chemokine receptor and the complementary receptor and Fc receptor which we have known. The complementary receptor, key receptor are CR1 and CR3, whereas chemokine receptor CCR7 and CXCR4. So we have known till now all the type of immune cell receptor. The first one is the PRR, which is equal to the innate immunity, and it helps in detecting the PAMPs and TAMs. Then BCR and TCR, they are the adaptive immunity. In that it recognizes specific antigen and NK receptor. It balance the killing or sparing of the cell. Then FCR, they bind to the antibody and trigger the effector function. Then chemokine receptor, they guide the cell migration, and complementary receptor detect the opsonized particle. So there is a complete overview of this type of immune cell receptor. We have known all of them, and so we have known the immune cell receptor of the immune system and how to detect and decide and response. So it was a revision video for the immune cell receptor. So those who have gone through the book, it is very easy for them to understand this video. Thanks for watching. Do like, share and subscribe.